what I'm going to do is I'm going to actually focus on 2 or 3 use cases of using data, just to give you a feeling for some of the things that are actually possible now. I just also want to emphasize things are changing quite dramatically as well. And things are changing locally for us. This is the town where I live and work, and this is the sort of new developments that we have. This is a new museum that's just been built. So look, things are changing around us all the time. So what I'm going to do is I'm going to, until quite recently, the standard paradigm in plant genomics was that we'd invest in model species genomes, and these would provide foundation to underpin genomic and genetic advances in crops. So we'd sort out all this sort of hard stuff in the models, and then you'd be able to get it in crops. And this was based on the concept that there was a conservation, for example, both in terms of gene and gene function, but also in terms of chromosome segments. And you could anchor a set of genetically resolved traits in cereals, for example, to sequence-level information in rice and brachypodium. And although this approach has been of fantastic value, it's rapidly being superseded because of progress in sequencing and assembly technologies that enable you to do a lot more directly in the crop species. But also, one really important thing that's part of this is that there is still a fantastic opportunity from the comparative work, because again, you can learn a lot about things by comparisons across species. And some of the examples I'm going to give are really trying to focus on some of the benefits from that. I'm going to walk through an example with oil palm, and again, if Rob Martinson had been here, we could have, I could have shown this, because I started doing this little exercise, and this work was done one evening, because I was invited to talk at the major oil palm conference in Malaysia. I have an interest in oil palm as well. And I was asked to talk about our barley work, and I thought, well, it's going to be a bit boring for oil palm people. I wanted to get some connectivity. And this happened just after the oil palm sequence had been published and the oil palm shell thickness gene had been identified. Now, again, I have an interest in this. I'm one of the authors on a long time ago, the RFLP map that first of all located this into the oil palm genome. Anyway, from the Nature publication, basically it was a Mazbox gene encoding a homologue of seed stick, which controls ovule identity and seed development in Arabidopsis. There was a clear and obvious rice homologue, so there was an obvious homologue into the cereals. One of the things is that finding homologues can be a little bit awkward with Mazbox genes, because they're driven by the high homology in the actual Mazbox themselves. But what I want to do is walk through just some of the kind of connectivity you can do. Again, just to show I'm sort of recognized an informatician now, but we get out into the real world as well. So this is amongst oil palm, and Rajan Aju and Maya are some of the key people in Malaysia who are responsible for the oil palm sequence. Also, those people who worry about managing genetic resource collections, you ain't seen nothing when you try and manage a genetic resource collection in oil palm. You don't store seed, you store very large palms, which is something of an issue. Anyway, so we started off with the oil palm sequence, and what we did first of all was looked against the sequence resources for barley we have. We ran a blast analysis, and we get a whole range of different Mazbox genes. This is all the homology in the Mazbox, but the one we were interested in was the top one. We could right away find out where it mapped in barley against a number of populations. We could check, for example, against the oligos from various microarrays that we have to see whether we could actually find it on microarrays, and we could again just have stored blast comparisons to do reciprocal blast back as well. So we think we've got a clear candidate in barley for something that's probably the equivalent gene. So what can we do about it as well? So, the other thing is, again, we can say, well, where is it expressed? Again, we have a nice set of replicated RNA-seq data that we can go to and say, well, we have a range of different tissues, nicely replicated. So we actually could work out it's expressed in grain development as well. So again, we have something which looks like it might be an interesting gene even in a barley context as well. We can look a little bit more about it. We can just say, well, this is RNA-seq reads mapped onto the genome, and we can just check the genome structure and tron exon as well. So we can learn a lot about this. And this is like 10, 20-minute job to do this if you can connect it all up really quite nicely. We can then say, because we know where it is in the genome, we can say, well, do we have any sort of data that might say that, it might be an interesting candidate gene? So we have a set, so this is done again some time ago, so a few years ago, we have a set of 7,000 SNPs on 1,000 elite commercial barley lines, which we can do association analysis with. So again, we have this stored QTL data, and these are QTL confidence limits against one of the barley chromosomes. This is a whole range of different lines, so this goes down to 1,000 lines, someplace down here. And we know that actually it's the genome region corresponds to these QTL here roughly as well, so we thought that might be really quite interesting to look at. So we can take that and we can unselect all of the SNP markers, and we can just select for the markers that are actually under the confidence limits of the QTL, and we can sort for the haplotypes as well. So you can say, well, yeah, there's interesting haplotype performance in this area. So again, a few years ago, we were pretty much stuck at that, and we could go back and do some follow-up sort of directly, but we couldn't really mine information. We can do a bit more than this. So the first thing is also I forgot to say, the QTL is mapped by GWAS in this region control grain shape, size, and quality as well. So we thought, well, yeah, there might be some interesting connectivity there. But actually, there's now quite a lot of sequence diversity data out there that we can actually go to. So again, because we know what the gene is in rice, we can have a look at the Rice 3000 Genomes project, and we can essentially mine the sequence diversity for this gene in rice. And again, the IRIC database has a nice portal for it. We can stick in either a range in the genome or an individual gene and get the SNP variation across these 3,000 genomes. And again, they'll download it into Flapjack, which is one of our tools where we can play around a little bit with the data again. So this is we've now selected the equivalent gene in rice. We've exported it into Flapjack. This is just the top of the 3,000 genomes here, and these are the SNPs that are within the gene. We can then sort them by similarity, so you get essentially at the top of this heap, there's a whole range of things that have the same haplotype. We can color it by similarity just to get uniformity of haplotypes, so you can see what a haplotype looks like. We can move further down this stack, and we can find actually there's a different haplotype here. And we can look, if we can sample, this is the total stack here, there's sampling this bit here of the 3,000 genome stack, and we can see the haplotypes nicely lined up as well. So again, we've gone from information that's a potentially interesting gene, and we can identify a range of haplotypes that might be worthwhile exploring. So again, we could do this, we've been able to do this for some time now, but we're also starting to be able to do this in barley as well, because we've got exon capture data now in barley. Again, I'm just looking at a subset of the data we have. We've got a subset of 450 barley European varieties that span the history of UK relevant germplasm. And again, we can look at the diversity in the seed stick or ortholog now in barley. Rather than looking at genome area, we can actually look at the sequence variation of the gene. And this is some of the exome capture data. It's a little bit more patchy. The gene SNPs are really in the central region, but we also capture a little bit outside the area on either side as well, which may be interesting for regulatory regions as well. And again, we can do all of this sorting and looking at stuff here, and we can sort it. And again, you can see everything looks like it's got a fairly uniform haplotype here. Again, we can color by similarity to the top one. Then we can start to see, well, actually, there's not much in the way of variation in barley, and the variation up to this point is purely in the flanking sequences. It's not in the gene sequence itself. If we go down to the bottom of the stack, we find actually there is a little bit of variation within the gene, but actually, if you look at the gene model against it, it's actually in one of the introns of the gene. And actually, although the interesting thing about this is in very old barley varieties. They're even older than I am. So this is, the modern germplasm doesn't have any variation at all for this gene. So it's probably not a really candidate for, at all. But anyway, you can do all of this as a thought exercise. You don't have to waste your time, with a lot of effort. It's there, it's stored information. Okay, again, let's work through another example now. We have an interest, as many people do, in nitrogen utilization efficiency. And One of my colleagues, Luke Ramsey, had some data on barley and on wheat on an NUE, and he also knew that there was some QDL effect towards one end of chromosome 6 in barley. And so what we thought, well, we start to say, well, is there anything interesting that might be relevant there in terms of NUE utilization? And it turns out, in fact, yes, there is. Again, this is sort of a diagram of some of the key genes that are involved in nitrate uptake and utilization. And there's one particular class of transporters, which are the NR2, which are high affinity nitrate transporters. And these have been very well studied in both rice and in Arabidopsis. And in Arabidopsis, there's a couple of them. There's a couple of them in rice as well. And there's a lot of information about them, about the cell biology, how they interact as a dimer. And at the time, we were also working with colleagues in Ireland. We have a Lolium perenni genome as well. So we thought, well, let's have a look and start to have a look and see what we can find out in various cereals about this set of genes. Well, the first thing that we can look at, but sort of very crudely, is from a synteny point of view. We find that, there's actually, there are genes present in the equivalent positions, for example. So these genes are at one end of barley chromosome 6. That corresponds to a region in Brachypodium 3 and rice 2. So we've got synthetic genes here. Can we find out a bit more about them? Well, the fun thing is that there's a lot of copy number differences across the cereals when we look at it. So Arabidopsis has 2, rice has 2. It starts to increase. Sorghum has 3. Brachypodium has 4. Barley has 8-ish. I say that because there's some issue around the actual number, and we'll come back to that. Lolium has 11. One interesting about Lolium is that while we had our own sequence, and just before we published it, somebody else published another Lolium sequence, and their annotation said there were 2 genes in Lolium. And it turned out that they were actually using comparative data with rice to essentially structure the Lolium data. And because there's only 2 in rice, they said, well, actually, there's only 2 in Lolium. So they had no indication of the other, of the other 9 that were there. It starts to get really interesting in wheat. In wheat, there's 43 of them in hexaploid wheat. There's 18 in rye, and again, if you go into some of the other monocots, there's only 1 in yam, there's 1 in oil palm, and there's 5 in foxtail millet. So there's a lot of variation. In barley and wheat, they occur in the genome of roughly a group that's roughly again several hundred kb in size. In rice, it's over a very small region. And these are highly conserved genes. There are single exon genes as well, which makes actually annotation of them relatively easy. We can have a look at these relationships in terms of the phylogeny of them. So if we look at, for example, in small grain cereals and grasses, there is a major duplication that's ancestral to them, and so we can actually see that there are 2 major groups. So this a big chunk of them divides into 2, and it structurally divides into 2, although they're relatively close together in the genome. What we also know is that even within that grouping, within different lineages, there's been significant expansion. So for example, the Lolium ones are actually expanded within Lolium separately from the expansion that's happened within the others. I think in the case of barley and wheat, you quite often get these little clusters that have 1 barley one and 3 wheat ones in them as well. So there's at least some parallel indication here. Separately, if you look at the panicoid cereals, there's actually fewer of them, but again, there's a duplication which is separate from the one that occurred in these. And but also foxtail millet actually has its own little expansion as well. So we've got lots of stuff going on here. And again, we have stored gene expression data. These things are all incredibly highly root-specific in expression. We can actually work out which ones are the most highly expressed. Again, this is without doing any new work. And again, we can start to look at diversity for these things as well. So this is again going to the rice 3000 genome data, but this is the structure and where they occur in rice. So these are the 2 genes here in rice. There's a retro element between them, and there's a couple of genes on either side. So we can look at this region in terms of the rice 3000 genome set. So again, this is this area in rice 3000 genomes. Again, it's a sort of long way down here. These are the two red ones here, are the two transporters, this is the retroelement in the middle, and these are the genes on either side. So again, you see it's a bit patchy, some of the data, and again, it's nice looking at sequence data in this sort of context, because you can see where the information, and you can see some interesting features if you look at it here. So we can start off, we can unselect all of the genes, we can select the SNPs associated with one gene, and just to show this is the pattern across this region. And you can show that quite nicely. And again, you can look at, this is all of the 3,000 ones. So this is the stack here. I've divided the stack into 4 so we can squeeze it onto the screen. So what I've just simply done is sorted it into haplotypes. So there's a relatively limited, but again, the gene that's on the end actually has presence-absence variation, as does the retrotransposon as well. So again, we can find out a lot about what's going on. Again, we can go to Barley, to the exome capture data in Barley, and look at it. So in Barley, in the region, there's 472 SNPs, and we have this across, in this data set we're looking at here, 466 Barley varieties. We've just produced recently a new Barley 50K chip, and that's got 9 SNPs in this region. So it's, this just gives you an idea of the difference in resolution. And the SNPs that are on this array are actually in this capture set as well. So you can see exactly the same SNPs. So again, just to show you, this is one of them here, and again, we can sort this into haplotypes. It's a little bit small here, but you can see that there's the majority of them actually have an identical haplotype, and for this one here, and there's a little bit of variation at the end. And we can go across this set, and again, remember, we've got multiple ones here as well, looking at this. When we come to this one in the middle here, it looks like there's a lot of variation. There's some weird shit going on, is what it looks like as far as we were concerned. So we thought we'd have a look at this in a little bit more detail. And so I've just expanded out a bit in this. And when you expand out a bit in this and you look at it, and you see, in fact, there's a lot of stuff that's heterozygous. And this is Barley, it's heterozygous. Come on, what the hell's going on? So we can look at this, and again, just to emphasize it, again, we can just highlight the heterozygotes. And if you actually go down and look at this at the read mapping level, what you actually find is that the reference sequence in Morex looks completely homozygous, but it's when you look at other lines, in fact, you see evidence of what is heterozygotic, and it's not really heterozygotic. What it is there's probably 2 genes here, and we're getting reads mapping from 2 genes. So we can recognize that, again, in Barley, there's actually variation for copy number. So I've left one gene off the end of this because, again, this one was a little bit different, and when we looked at it, there's something a little bit strange going on here. What we find is that there are all these sort of regular gaps down it, and when we looked at the SNP data for that. So again, this is the Morex cultivar at the top. So this is the reference sequence that everything's been mapped to at the top. There's a big chunk of stuff that looks exactly the same as the reference sequence, but there's a big chunk of stuff that has half the genes not there apparently. So again, what's going on? And we can find out that quite easily if we actually go and look at some real sequence. So this is the Morex reference sequence. So this is another sequence of Barley. And what we find is actually Morex had this gene fusion event, and it's lost a gene in the middle, basically. So we're getting this strangeness going on. Okay, so let's we'll say that advances in sequencing genotyping technology are providing new opportunities. Rather than the model versus real crop paradigm, I like real versus as opposed to models. There's actually a great deal of value in looking at genome evolution across species. So you can, by putting things together, you can get a lot more. And there's a growing body of molecular diversity data that can be efficiently mined. There's a lot you can do before you have to spend a lot more time and effort doing the difficult stuff and the really interesting stuff, which is the actual biology as well. And again, increasingly, as people have been highlighting, this idea of a pan-genome reference is becoming really important in terms of understanding patterns of variation. And also, just to emphasize, we need to take care in how we interpret diversity as well. And actually, this is the pan-genome view that I like. So this is the tube map view that comes from the VG group on the human side. And so basically a genotype is a trajectory through this graph. And I think it's a really nice way to look at this. I mean, it's nice in terms of being able to structure it, but also increasingly it looks like it's a really very sensible way to be able to to handle read data, to map it onto a pan genome, and also then to be able to store the information potentially at a haplotype level rather than an individual SNP level. Again, it reduces some of the storage burden a lot. Okay, we've got a little bit of time in the end. I'll do some acknowledgements and jump to just a little bit at the end. This is the group that I mostly work with. So Ian and Gordon, who do a lot of our major software development. Linda and Ian, who do a lot of the sequence and SNP handling work. Paul and Sebastian, who do a lot of our database structure and design. We've had lots of input from friends that have helped us, to identify issues, and we work closely with a lot of people at CIMMYT who have been really enjoyable people to work with. Over the last 2 or 3 years, we've increasingly worked with the Gobi group and others at Cornell, which we found really stimulating. Obviously, we have a great deal of contact with the genetics group at James Hutton. The Barley work is incredibly dependent on the International Barley Sequencing Group, and Jesse Kennedy at Napier has been really influential in some of the computational stuff we do, and also we have a range of funders. So I've got a little bit of time just to pick up on various things that I've heard through the meeting as well. So again, somebody talked about, can you see the 1B 1R translocation? Yeah, you can really see it really easily if you've got SNP data. So this is some SNP data from Matthew Reynolds at CIMMYT. So you can take that again. You can do the the old sorting bit and coloring bit. So this is a 1B chromosome. You can essentially identify a chunk of it where you'd expect the 1B 1R translocation to be. You can actually sort it, and you can see the sort and the structure. And if we color up differently, you can see where the translocation is really quite nicely. It shows up. Okay. So again, I think Mike Bevan was talking about how related some cultivars are in the UK as well, and again, we can see that quite easily. We do quite a lot of visualization now on a pedigree graph as well, which is quite fun as a thing. So this is the 2016 recommended list for barley in the UK, and it all links together in a quite small pedigree graph as well, and we can just color up by the different breeding companies as well, so you can see where the different companies are, because they all have crossing arrangements. So it's and in fact, a couple of years ago, we had a situation where we had 3 barley lines on the recommended list that all came from the same cross done by different breeders. It's that close by. And again, you can use this pedigree graph as a way of laying on information. So this is just the present absence of a really interesting gene that is involved in relevant to whiskey production, which is very important from a Scottish point of view. We can make much bigger pedigree graphs. So this is probably a lot of the background to the UK barley cultivars. And again, we can link them all really nicely in a graph. And we can highlight, for example, we can make them big by the number of times they've been used as a parent. So you can see the dominant varieties that have been used as parents really quite nicely in this. And we can do similar things with wheat. This is the UK winter wheat recommended varieties and pedigrees. And this, I think, was last updated, last year. And so, again, you can see they're all sort of highly related. We can color them by which breeding company bred them. And you can see, there's lots of interaction. We can color them up by the quality group that the various wheats belong to. And again, you can see they're all really quite closely interacted. And the other thing we can do is we can identify how often things have been used as a parent. And you can see why Mike Bevan is sequencing Robigis, because it's pretty important as a parent in the current germplasm. Again, we can use some of our tools to look at lots of different stuff. So this is maize data. This is a recent publication that came from INRA on diversity in maize. Again, we can color up various groupings within this, and we can use this to browse and query and play around with the data a little. This is maize from CIMMYT, and this is maize in Scotland. We can grow maize at 56 degrees north. It's really impressive. This is climate change in action, really, I think. Okay, so where are we going from here? Just very quickly at the end. We're interested in integration of tools, and so, for example, because we have a number of visualization tools, we can integrate, or we're working towards integrating across them. So you can select in one tool, for example, and look at graphical genotypes, and you can see the pedigree relationship and see that live and joined up. You can identify local haplotypes at a database level and paint them across. One of the things we're doing is working with a number of colleagues around the world to develop a common API for a lot of breeding tools. And one key element in this is CGR Excellence in Breeding Platform, which I think is going to be really important. The other thing that we're doing as well, although our primary interest has been in terms of using analysis things, we've not been developer of analysis, but we now have a collaboration with Biometrics and Wageningen and VSNI, really to develop essentially analysis pipelines to support plant breeding efforts. And this is really covering everything from environmental sensing to experimental design to handling and developing marker information, to handling trial data, to doing QTL or predictions, and doing decision support. And we feel visualization is important because people need to get and look at data and make decisions really quickly. Scalability is becoming really important, and linking things together is important. Okay, thank you.
Well, thanks a lot for giving us 1.5 talks and staying on time. Questions?
Fantastic presentation. It's I think some of your software is the best software to use for this sort of work. You got some really good information on pedigrees and genotypes. So have you gone through and mined that information to identify alleles which have been predominantly selected in particular environments? So can you look to see the impact of the breeding program on allele frequencies?
We haven't, but that's clearly a possibility. That's really a possibility. I mean, we're at the stage now where lots of information is coming together, so I think there are all sorts of possibilities now. Yeah.
Always. With regard to the high affinity nitrate transporters, have you analyzed Maize too, or I don't see Maize there?
Yeah, Maize is in there. Yeah. Well, we can tell you a little bit about Maize. There's not so much diversity. There's only a limited number in Maize. I mean, one thing I didn't really, because of lack of time, I was trying to obviously push a lot through quickly, is that these particular transporters work as a dimer with another transporter. And there's only one of the other transporter, and one of the really interesting things we've not really looked at yet, but is whether there's any allelic diversity in the other part of the dimer, and whether it corresponds and matches up with the diversity in the ones that we've looked at. Okay.
If I understood it correctly, then you also integrate RNA-seq data, right? So do you also compute and provide eQTL data, for example?
Yes.
And what about alternative splicing, if I may ask, or alternative 5' ends?
I haven't gone into alternative splicing, but just as haplotype diversity is, alternative splicing is becoming stunningly important. In fact, I share an office with the person who does the analysis for us now. We have in John Brown, based in the Institute, a long-term expert in alternative splicing in plants, and having teased John for years about there's all this, alternative splicing he's been telling us was going to be really important, and we said, well, yeah, when is it going to be really important? Well, it's really important. We can tell you that's the case now. They started off with Arabidopsis work, and they've essentially almost doubled the number of gene models in Arabidopsis based on alternative splicing. And one of the things that they found when you do that, and then you map the reads back onto a very full description of the transcriptome, you get quite big differences in the expression levels that you're able to calculate. Things like the length of the UTR and other things make a big difference in terms of read-based expression analysis. So, alternative splicing is important. There's lots of really nice data coming out now which show that you get splicing variant switches in various stresses, but also it's really just important just in terms of quantifying it. And one of the things that we're trying to do with Barley and other things just now is build up new reference transcripts in order to improve the quality of that. Okay.
Well, thank you very much. Thanks for joining me in thinking.